Hi, my name is Steve Pressé, and I'm faculty in physics in the School of Molecular Sciences. Let me go ahead and share my screen so you can see my presentation. Now, it always takes a second to start. And here we are. So my lab is focused in three broad directions, one of which is statistical mechanical theory and modeling. And in this particular case, we're interested in hydrodynamics at room temperature and the transport of micron-sized particles such as organelles within fluids. Another broad area of focus relates to questions of inference from mostly in vivo data. In other words, from the rawest form of data, we try to infer the rules of life. And finally, I'll tell you a little bit over the course of a couple slides about our interest in experiments and unraveling bacterial hunting behavior. In other words, bacterial predator-prey dynamics. So on the theory front, and in particular in the inference portion of the lab, we have a team of mathematicians, physicists, and chemists who start from the rawest form of data. In this case, these are photon arrivals, process them through our means, through our mathematics, our novel means, and ultimately arrive at rules of life. For example, the assembly, disassembly of protein complexes, transcription, and other intracellular processes. Now, as with any new area of physics, as we are trying to unravel the rules of life from individual photon arrivals, we do need to develop novel mathematics. So this area of the lab, or this area of research, is really targeted at people who enjoy mathematics, who enjoy stochastic, or who might learn to enjoy stochastic processes, and who enjoy programming as well. As I've mentioned, the ultimate goal is to move away from one-body physics, which is what we're very good at in monitoring, say, the behavior of a single molecule within a cell, and ultimately to arrive at pictures of interacting molecules and understand them from the rawest form of data that is available, reporting back on their behavior. Now, on the experimental front, we are very interested in a little bacterial predator, here shown in yellow, called BB, which is short for Delovibrio bacteriovorus. This little predator bumps into prey. It appears at random. Once it does, it burrows its way into its periplasmic space, invades it, replicates, and then ultimately releases progeny back into the open. Now, what's particularly interesting about this predator is that despite the many eons over which it has existed on Earth, the best explanation we have for how it locates its prey is that it bumps into it at random. But little by little, my lab has unraveled rules of hydrodynamics that ultimately facilitate its ability to locate prey. And what I mean by that is that we ultimately learn that these predators exploit passive hydrodynamic forces in order to localize themselves to surfaces. When they do, one artifact of them exploiting hydrodynamics is the fact that they make circles or they get trapped around circular defects. But since prey do a very similar thing, they've essentially reduced the dimensionality of their search for prey from bulk fluid in 3D to these surfaces where they are much more successful in bumping into their prey. So this ultimately leads to future directions in the lab where we'd like to be able to map out hydrodynamic flow fields around bacteria as well as interacting bacteria. Now, another interesting thing is that this little predator has one flagellum, that little tail that it uses to propel itself in solution. And occasionally it'll get stuck by its flagellum onto the surface and sort of rotate in the following way. This highlights the point that bacteria have to expend a high amount of energy, especially predators, in order to locate their prey. And the question is, how do they apportion or expend that energy in difficult, challenging environments? In other words, when prey are not present. And what we're exploring and what we'd like to be able to continue exploring with incoming graduate students are complex motility strategies where the predators seem to alternate between two modes of motility as a function of time elapsed since they have received clear signal from prey. Well, I'd like to thank all of you in attendance. My email is here. It's spressé@asu.edu. We have a large number of experimental collaborators when it comes to our theoretical work. As I mentioned, we have a team of mathematicians and physicists. as well as chemists, all working towards unraveling the rules of life. On the experimental front, as well, we are recruiting and very much look forward to meeting you here at ASU. So let me now stop sharing and thank you all for attending.